PhD, is the founder and CEO of InSilico Medicine, a leader in next-generation artificial intelligence technologies for drug discovery and biomarker development. He is also the founder and CEO of Deep Longevity Incorporated, a global company developing a broad range of artificial intelligence-based biomarkers of aging and longevity. Since 2012, he has published over 130 peer-reviewed research papers and 2 books, including The Ageless Generation: How Biomedical Advances Will Transform the Global Economy. He serves on the editorial boards of Aging Research Reviews, Aging Trends in Molecular Medicine, Frontiers in Genetics, and co-chairs the annual Aging Research, Drug Discovery, and AI Forum, of which there is the 7th annual in 2020, at Basel Life, one of Europe's largest industry events in drug discovery. He is the adjunct professor of artificial intelligence at the Buck Institute for Research on Aging.
Before I start sharing slides, I'd like to thank the organizers for inviting me to speak to you today. So Greek Pharmaceutical Students Federation is a wonderful organization. I looked them up, and Greece gave birth to such prominent people and pharmaceutical executives as Albert Bourla, for example, the CEO of Pfizer, one of the largest companies in the world, and many others, Seraphim Bartzglow, the famous professor of AI at Stanford, and now he is the chief data officer at one of our kind of quote-unquote competitors that we don't consider to be a competitor, called Insitro. And from what I've heard, Pfizer is planning to start an AI center in Thessaloniki, which is very amazing. And to me, that's great news. My heritage is, as you have guessed from the family name, Slavic. So I was born in Latvia, got my first 2 bachelor degrees in Canada, so I'm Canadian by citizenship, and worked in the semiconductor industry for a while, then made some money to be able to pursue research, and decided to go after humanity's biggest problem, aging. So from the introduction, you've probably noticed that aging was mentioned multiple times, and if you are in pharmacy or medicine or anything related to healthcare, your job is essentially to figure out a way to extend human life, regardless whether you're trying to address a disease, something that is very pressing, or turn it into a chronic form, or prevent disease, or preferably extend human productive longevity. So my talk today is going to focus on kind of 2 aspects. One is going to be in silico medicine and some applications of AI with some practical results. And then I'll switch gears into longevity and talk about the future of where I see the medicine going and pharmacology going, where you get the feedback from the various digital medicine tools and various diagnostics for the drugs that you create, faster than before. So for those of you who are interested in, working for a company like InSilico or InSilico, I highly recommend getting a very well-rounded background. So you need to know your pathways. So one of the most common interview questions I would ask is, draw a Wnt pathway or EGFR pathway or any other common pathway, like Notch pathway. If you don't do that, that means that you spent the last few years without, a lot of practical knowledge or theoretical knowledge in cell biology, and that means that you're not going to do well in the company. I might ask similar questions about chemistry and the various interactions of the very famous small molecules with the famous targets. And of course, I would ask about the various pharmacological effects of the very known drugs. And I might show you molecules that you will look at, and I will ask you to describe those molecules. So that's kind of the very bare minimum. The way we hire AI guys is usually through competitions. So we do a national competition, first virtual, and then we do on-site competitions for 6 days, trying to replicate and outperform one of our recent papers or somebody else's papers, like, for example, DeepMind's papers. And then we hire some of the best students or scientists who outperformed in the competition, and then we train them in chemistry and biology. So that's how we do things at Insilico. We try to work very, very hard. So, you know, you don't want to see my Sunday schedule, and all my team is working right now. And we try to work with the very, very high-level people, top experts in the industry. So now you probably can see my screen, correct, Tatiana?
Yes, yes, we can see your screen.
Yeah, perfect. So anytime you want to kind of read about what we do, I highly recommend going to Scholar and, you know, looking up some of our recent papers in AI. Our company does publish from time to time, at least the direction where we're going, and some of the famous papers that you might want to start with would be, you know, this Nature Biotechnology paper that I'm going to be talking about just briefly. But that's not the highlight of what we did in chemistry. Actually, another really cool paper that you might want to look at is this one. So entangled conditional adversarial autoencoder for de novo drug discovery. Highly recommend diving into that one because that's the first experimental validation of what we did with generative adversarial networks. And if you are an AI, highly recommend this paper, Reinforced Adversarial Neural Computer for de novo molecular design, and Adversarial Threshold Neural Computer for molecular de novo design. If you are in target discovery and, you know, you want to look at some of the more classical papers on target ID, I highly recommend going after this paper. So use of deep neural networks ensembles to identify embryonic to fetal transition markers. So we identified COX7A1 as a promising target in cancer and also in regenerative medicine. That was before TensorFlow, etc. We published it, went online in 2017, but this work was done in 2015. So without further ado, I'll share my presentation and go through the slides. So as I was generously introduced, as per the introduction, I am the CEO of a company called Insilico Medicine. So it focuses on end-to-end drug discovery. So we look at pretty much everything from identification of novel drug targets to prediction of clinical trials outcomes. And I'm also currently the Chief Longevity Officer of Deep Longevity. It's a company which is focused on biomarkers of aging. It was just acquired by a public company called Region Pacific in Hong Kong. And that's why I have to make this disclaimer. So this is not an investment advice, and we are not making any forward-looking statements because it's a public company now. What we're focusing on in both companies is a technology called deep generative reinforcement learning. So you have probably seen deep reinforcement learning in, you know, AlphaGo, in Go competitions, in various video games that DeepMind has outperformed with their technology, and they were really kind of the true pioneer of deep reinforcement learning. So this is the technology that helps you learn strategy. And generative adversarial networks, the technology that I'm going to be talking about, it allows you to create new objects with AI with a defined set of properties. So think deepfakes, but you can use it for many, many applications. And if you combine those two technologies, basically AI imagination and AI strategy, you can generate certain structures, certain objects, certain data with the desired objective in mind. So we use it for many purposes, and we try to build multi-purpose models that can be applied for multiple areas at the same time, primarily for kind of biological age prediction and monitoring. That's Deep Longevity. And if I kind of show you what we do at different companies, so Insilico Medicine uses this technology for biomarker development, biological target identification, Molecular screening, we're quite famous for molecular generation, so you can generate molecules with specific desired properties. We also use it for clinical trial outcome prediction. And for deep longevity, we use it for predictions of age, of human biological age. I will talk about that today briefly. Biomarker development, target ID as well, but for aging, real world data analytics, and we even started going into kind of mental health, depression, motivation, and behavioral modification. So that's just this few last months that I'm interested in psychology, and we're using this technology quite a lot to understand human behavioral patterns as well. So now I am a student. My clicker doesn't work. I have to use manual. So I'll be talking a lot about what we can do with generative adversarial networks. But for those of you who are new to this technology, I highly recommend this paper by Scott Reed, published in mid-2016 as part of ICML conference. That's International Conference on Machine Learning, probably #2 in the world. #1 is NeurIPS, previously called NIPS, Neural Information Processing Systems. Those are two top conferences. And in mid-2016, they put this paper out to demonstrate the applications of GANs. And you can see that there is a generator network which is trained on millions of images to generate meaningful noise in response to data to some generation condition. In this case, the flower has small round violet petals with dark purple center. And there is a discriminator network which captures the output of the generator and tries to compare it to the ground truth and tries to capture whether the output of the generator is true or false. So those two deep neural networks, they compete with each other. One is trying to trick the discriminator, and the discriminator is trying to figure out whether the input from the generator is true or false. And as they compete, they get pretty good at that. And again, 2016, that was pretty revolutionary. When you give the generation conditions, so something that you want in an image, like this small bird has a pink breast and crown and black primaries and secondaries, and the deep neural network imagines those images, and you can see that many of the generation conditions are met, right? So you've got a small bird, pink breast, crown, black primaries and secondaries. So AI imagined those birds. If you were to do this yourself, you would not do a much better job actually at imagining those birds. And technically what we're doing is we're sampling from a distribution of those images that the deep neural network came up with, right? And you can see that it does pretty good job. And again, this is 2016. So to me, it feels like ancient times, even though only 4 years have passed. And in 2017, you could already start generating very good data with very realistic faces. For example, this is called deep fakes. Those people do not exist. They were generated with specific generation conditions. And I can show you how we do it, for example, on images with many, many different conditions. And you can generate those faces in kind of many modes. You can either start with a template. So think this is very important. So you start with a template. For example, you give it Brad Pitt, a picture of Brad Pitt, and you say, give me this Brad Pitt who is older, who is now African-American, and who is now female, and is wearing glasses, and is bald. And the deep neural network, GAN, will generate a Brad Pitt look-alike with those additional properties that it learned from the millions of other images. And that's basically template-based generation. Or you can ask trained GAN to generate a male with those 4 or 5 properties, and it will generate the whole distribution of those males that will look very differently, and they will not look like Brad Pitt, but you will see many of those specific conditions met. And that's basically continuous. Pictures are continuous data types. If you're talking about molecules, molecules are discrete data types, right? So here, one pixel change, or maybe if you even close the eye, you will still recognize it as a person with all of those conditions that you want. And it will fit your requirements in many cases. But if you're generating molecules, molecular structures, one atomic substitution or one weird double bond will screw up your molecule dramatically, right? So it will not fit into the pocket, it will be toxic, etc. So one atomic change makes dramatic differences. So you need to be much, much better at generating great output with the desired generation conditions when you're doing this with chemistry. So if you want to learn a little bit more about this technology and the applications of this technology to either data generation or generation of novel molecules, just go to PubMed and do in silico plus medicine. You'll get the papers here. Just take a screenshot. Those are some of the most representative papers before 2019. And I'll talk a little bit about in silico now. So in silico is a company which is specializing in target discovery. So we identify novel targets. We do generate novel molecules. We use this generative adversarial networks and deep reinforcement learning that I've mentioned. We have many papers. Actually, this is an old slide. Now we have more than 120 paper records as in silico medicine, many, many more papers that other people produce using our algorithms. We are usually the originator of the IP, so we invent it. I try to work with very creative people who don't think about the limitations. So when I'm asking them to perform a miracle, they usually say yes. And this work that we've done on GANs for small molecule generation that was done in 2016 was highlighted as some MIT Technology Review's top 10 breakthroughs of 2020. We're currently about 130 people worldwide. 80 of us are AI scientists. I'm calling in from Hong Kong. That's the headquarters. However, we have only a few people here. We have an office in Rockville, so at the campus of the Johns Hopkins University in Rockville. We have an office in Shanghai at JLabs, the campus of the Johnson & Johnson company. Taipei, right next to 101. We also are in Moscow and St. Petersburg, so it's very easy to fly to Greece if necessary. We raised 52 million since inception, and currently there is no shortage of investors who are interested in funding us. So it's very nice to be us. If you are, most of you are familiar with this slide, since you are students of pharmacology, and this is the seminal paper. But just to refresh your memory, this is Steve Paul from Eli Lilly at that time, published a paper in 2010, How to Improve R&D Productivity, the Pharmaceuticals Industry's Greatest Challenge. And he showed the various phases of drug discovery with the probability of success, so this PTS, cycle time, so how long it takes, cost per launch approximated in millions. And that was 2010. In 2013, the overall cost of drug development exceeded 2.6 billion. So it's a very costly process, takes 10 years, and that doesn't even include target discovery. So target discovery is the most complex process, I think, in the pharmaceutical industry. Identifying a great target and validating it in the context of the disease, it may take you, seconds. Or it might take you to decades, and it might cost more than 2.6 billion. What is important is that currently target validation is performed in disease models using animals or organoids or human cells, and the real validation comes in phase 2, right? That's where you test efficacy. So very often when you have discovered some hypothesis back in the days, right? So 10 years before you test it on humans, you only know whether this hypothesis is correct when you test it in human patients in a phase 2 clinical trial. And there are many other things that can go wrong with the trial design and other things. So it's not necessarily the target hypothesis, but you will be able to test it only in phase 2 10 years down the road. And that's the most dramatic part about drug discovery. it also shows you that even if you are amazing here, so target to hit to preclinical, and you can basically almost eliminate this phase. So think about as a Ferrari going from 0 to 100 very quickly, faster than any other car. After that, you move with the speed of traffic in later preclinical phase 1, phase 2, and you have the same probability of, well, hopefully less probability of crashing because hopefully your target hypothesis was better here. But at the end of the day, you are not dramatically improving the process. And that's also the curse of AI-powered drug discovery, because the ultimate validation comes in phase 2. So that is why it's extremely important to connect all of those pieces together. And what I am trying to do at Insilico and also at Deep Longevity is we are embracing MMA principles in drug discovery. Before MMA, people were arguing, what is more important, a boxer or a judo practitioner, who is going to win and who is a better fighter. And at the end of the day, people started competing in MMA, and many of those, taekwondo practitioners, boxing practitioners, they had to pick up wrestling, they had to pick up jiu-jitsu. You had to get great at both hitting and kicking and working on the floor. And there is no other choice. You have to be good at all of those disciplines, otherwise you're going to get beaten. And this is the real world environment, right? This is what you're going to face on the street. People are not going to follow the rules of boxing or taekwondo on the street. And this is what we are facing in the pharmaceutical industry and in drug discovery. You need to be good at all of that to be able to really deliver great products. So we are trying to go after pretty much any disease that is accessible by our algorithms. Currently, it's mostly diseases where a lot of multi-omics data is available, but we can now go just with text-based approaches for target discovery as well. You need to know pretty much every target discovery technique out there and need to combine them to be able to gain more confidence in your target and be able to go after novel targets. So there are very few novel targets out there that can be easily validated. So you need to figure out new ways to validate. And usually you validate by creating more target hypotheses and looking at where those target hypotheses cluster together. So you need to know many methods. You need to be able to bridge biology and chemistry, think about how to very quickly validate those targets in the most cost-effective and rapid way, so you don't waste a lot of time and you don't waste a decade to get to phase 2 human trials. So you need to be creative here, and you need to know how to create great chemistry, so to very, very quickly deliver great molecules, because in the pharmaceutical industry, as you know, the most value, the most power is in the molecule. So if you have this validated molecule that hits the right target in a specific patient subpopulation, you will be able to drive a lot of revenue. So if you patented it properly and if that molecule works, it's a gold mine, right? So that's what you want to get at the end, and you want to help a lot of patients, and you even need to be able to brag about your win, right? So you need to be able to talk about it, to properly present it. And I'm trying to train people at Insilico to be the MMA fighters in drug discovery. So they need to know both biology and chemistry really well. If you are just trying to specialize in one area, it's not enough. You have to really understand the entire spectrum and be able to converse with multidisciplinary experts. We are trying to basically integrate this in Insilico as a philosophy. So we do both target discovery, generative chemistry, virtual screening, clinical trials prediction with next-gen AI from ground up. Here is how we basically, it's on the website, so you can go and look at how we do it and what are the major building blocks we're using. We try to publish the papers that describe the very basic proof of concept of the direction we're taking. So if it's a pathway-based target scoring system module that we're including into the pipeline. We usually try to at least publish the pathway algorithm that we would be using. And for dimensionality reduction, what kind of dimensionality reduction algorithm we would be using. And then we would look at how do we prioritize different pathways, different targets. We would also explain, at least in broad strokes, how we do that. So it's very easy to kind of follow what we do, follow our trail, and either help us improve or join the company or invest in the company or partner with it. Quite unfortunately, many pharmaceutical companies don't do that. So they don't take the time to read those papers. Now they do it more often, but when we just started, it wasn't a general practice. So they want us to explain all this all the time in maybe one specific discipline, but nobody looked at us in a big picture. And if you want to look at GANs, for example, and how we apply them to generation of novel molecules, we also published. For prediction of clinical trials outcomes, that's kind of the holy grail of drug development. So you want to predict the success of a clinical trial study with high accuracy and also understand why it is going to fail, right? And especially when you are doing it for some high-profile project within big pharma, you need to understand that you know, hundreds and sometimes thousands of people worked on this program. $500 million, $1 billion, $2 billion have been spent before it reached you know, phase 2, phase 3. So when you are making this prediction and explanation, you need to realize that you know, a lot of experts have contributed to this work, and they usually know what they were doing. So it's very difficult to publish those predictions without explaining in very high detail how we do this and providing the code for peer-reviewed journals. So that's why we sometimes publish on arXiv. So either on bioRxiv or ResearchGate, just the predictions and general philosophy to explain how we are doing this particular approach, how we're approaching this, and what are the predictions and what are the expected outcomes. I'll show you, since, you know, let's do it a little bit less formally. So I'll show you how we do it. So for ResearchGate, for example, it's a preprint server that allows you to put articles there, date stamp them, and then ask other people to comment. So here is one preprint we submitted on you know, the Fool's Day, so April 1st, just so I remember. And it's called Multimodal AI Engine for Clinical Trials Outcomes Prediction Prospective Case Study for Big Pharma Q2 2020. If you go back and try to remember what happened at that time, right? So April 1st, that was currently, well, that's when COVID was classified as a pandemic. And a lot of clinical trials went south, right? They were either paused, discontinued, or primarily paused. And now they become less predictable because many trials got deprioritized. But here's what we did. So we explained how we use those engines to predict clinical trials. We took Novartis's clinical trials that were expected to read out in Q2. Now many of them were postponed to Q4 or in Q1 next year. We also explained the methodology, the data that we use, the different probabilities for the various disease areas for which we already have done retrospective validation or quasi-prospective. So when you train up until, let's say, 2010 and then test on, so predict those trials that are to read out from 2010 to 2018, for example, and compare actuals to predicted. So this is basically the stats. And then this is the most important table. We take predictions for the clinical trials that we're supposed to read out in Q2 and give the probability for this clinical trial. For example, here it's a small molecule targeting chronic squamous urticaria, phase 2, expected to read out soon. So it's in phase 2. So we are predicting phase 2 to phase 3 transition, right? And here for VP route, for childhood onset asthma, it was in phase 2. We are predicting phase 3 transition. So 0.11, that's very low. And by the way, it just got discontinued. And then we also show how we do this, right? So we have a trial choice score, target choice score. So for us, that's the most important score, which basically implicates the target in a disease. Trend score, so how trendy is the target. Drug structure score, if it's a small molecule. We don't assign too much weight to this prediction, but anyways. Trial design score, so how well the trial was designed. Those scores come from our AI. Oh, and text mining score, that even assesses the credibility of the scientists who were involved with the molecule early in the early preclinical days, and then at the clinical stage as well. So a lot of text mining scores actually are incorporated in this text mining score. And then we also show how the different features that went into the predictor contribute to the accuracy of the prediction, and is it positive or negative. So this is a technology called SHOP, which makes deep neural networks a little bit more explainable. So we make those prophetic predictions, and then we compare them to actuals. That's how we like to do it. Just wanted to show you an example of what I meant by doing prophetic examples and publishing on preprint. And then at the end of the year, we're going to look at how well we did with those predictions and compare actuals to predictions. And then we will just maybe either announce or publish. So the major customer for this is actually not pharma. So pharma doesn't want to know very often, especially from a company which is from the outside, and it's not McKinsey or one of the big consulting firms. The major customer for this are hedge funds who are betting on smaller biotechs that are very sensitive to outcomes of clinical trials. So if you are planning an alternative career, you might want to pursue a career in being an analyst in one of the big hedge funds betting on the pharmaceutical companies. And I think that there needs to be more of those funds, because very often those companies are betting on something that is too much of a me too, or there is not enough scientific base, and people are just carrying those clinical trials that need to be discontinued. So we also have a really cool project called MOSES, Molecular Sets. If you like to learn about generative chemistry, or play around with this in this field and try your hand at generative chemistry, you can go to MOSES. There are many models that we have implemented already, and you can see how they perform for generation of molecules for specific targets. So we basically try to bring our AI engine to the state where it can address pretty much any stage of the pharmaceutical R&D marathon. So we can do it either in a sprint format, and we also bet on our own molecules. So if you look at the history of deep learning, it goes back to 1940s, and the concept of deep neural nets is not new at all. And a lot of major seminal work was done in the 90s and early 2000s by those pioneers like Geoffrey Hinton, Jan LeCun, Yoshua Bengio. But only in 2014, the concept of GANs was proposed by a brilliant young scientist, Ian Goodfellow, at that time at the University of Montreal. And now, then he went to Google, and now I think he is with Apple, leading the AI initiative and also teaching. So this concept that I've just described, we use it everywhere. And we were, to my knowledge, the first to publish in a peer-reviewed journal showing the applications of GANs to small molecule generation. So in 2016, this idea sounded very crazy, especially to the pharma companies. And I was doing a lot of work in target discovery using different methodologies. So when I even proposed it to my team, they were like, okay, Alex, you know what, we might want to leave now because we don't understand the stack. And we are already halfway through with a target discovery engine, which is designed for something completely different. But we bet on this technology, tried it out, put some resources, and then we did a lot of theoretical work. In 2017, we started synthesizing. So we started ordering synthesis of those molecules that were generated in a kind of deepfakes model, modus operandi. And many people did not like that either. We decided to use chemistry contract research organizations that specifically allow us to synthesize any chemistry, and we chose WuXi AppTec. Some of their chemists looked at the molecules that we were sending for synthesis and said, "Well, guys, do you need some help because those molecules are weird?" And we said, "No, we don't need any help. This is specifically to validate, what is being generated by AI." And at some point in time, they came back and invested in us because they realized how powerful this technology can be. And WuXi is the greatest chemistry company in the world, 30,000 synthetic chemists. They perform contractual synthesis for pretty much every big pharma and do a lot of work in biology and other areas, clinical trials. So around this time, so around end of 2017, beginning of 2018, we performed many validation experiments where the molecules finally worked. And this entangled conditional autoencoder already contains the kind of synthesized JAK inhibitor, not a novel target, but we wanted to try something that we can validate. And then many papers showing validation in other areas. So one of the most famous papers that we've published that kind of probably received more publicity than it deserved, because some of our earlier papers were also very cool. That's Nature Biotechnology, where one of our partners challenged us to generate novel molecules with specific properties for an antifibrotic target called DDR1, very well-known target, with some already known inhibitors. And we used the technology called Generative Tensorial Reinforcement Learning System, which combines generative approaches with reinforcement learning. We synthesized, tested in human cells, tested metabolic stability, microsomal stability, tested in vivo in mice PK. So nice ADME properties were confirmed, published Nature Biotech paper, and we did all of this in less than 2 months. So usually it takes a big pharma company much longer. And again, it's done by AI, so that was a pretty cool feat. And now I have some unpublished stuff that is coming through. We actually did the same thing, but for a novel target. Also fibrosis, but it's not DDR1, it's completely novel target. We generated synthetic data for a bunch of fibrotic diseases using generative biology. So think deepfakes, but not on molecules, but on biological data like gene expression, protein expression, and other data types. Identified promising drug targets, generated novel chemistry, and performed in vitro and vivo validation in multiple disease models. And now we're thinking about which disease to take it into. So if you look at the kind of timeline, March 2018, you remember I told you that at that time our generative chemistry capabilities were still in their infancy, but we also started doing generative biology. We identified novel targets, generated novel molecules for them, tested in a big pharma assay through kind of an open collaboration. Took those targets back, so big pharma doesn't know the target. They saw the molecules. We created novel molecules, then tested in human cells, in multiple fibrotic cells and cell models. Tested in mice for NASH, so it's non-alcoholic steatosis, but we also tested it in CCL4 induced steatosis and fibrosis. We tested it in multiple other mouse liver fibrosis models. And then we tested it on several other models like IPF. So right now, in January 2020, we were ready to go into IND enabling, but then we decided to go after an alternative disease, and had to, at that time, you probably remember that's COVID pandemic, right? Right after that. So most of the labs were closed, and we had to look for alternative labs. So that caused a delay. But in general, 18 months from discovery of a novel target to IND enabling ready compounds. So that's pretty cool. Another kind of applied example is that, so if you look at COVID-19, there are multiple strategies that you can pursue as either target ID company or aging research company or small molecule generation company. So of course, we had multiple approaches ourselves. And one of the approaches that we were pretty successful with is the C3-like main protease project. C3-like main protease is SARS-CoV-2, one of the key proteins. So you can use, theoretically, an inhibitor at pretty much any stage, all the way from exposure to recovery. Yeah, so here are all of the SARS-CoV-2 proteins. C3-like protease is encoded by the viral genome. It is one of the core proteins responsible for replication. So if you inhibit it, it dies. It doesn't replicate. And we decided to use a generative chemistry approach back in January. Generated 100 molecules, put them on online, so for other people to synthesize. So I know that some are still working on those. But they are not telling us. Then we did in April another generative chemistry round. So it took us 4 days to generate the compounds with a different crystal. We got a much better crystal. Then you can go to YouTube and actually see how medicinal chemists, human medicinal chemists, look at the AI-generated compounds in 3D and comment on them and see how they can improve them. So that's a very cool way to learn. And then we put those compounds out for synthesis as well. So some people are synthesizing. And then we decided to do it ourselves as well. And by the way, those are representative molecules for that generation that we did with Nanome. For the 6W63 crystal, you can see that they are sp3 rich, they are diverse, and medicinal chemists, when they look at them, they do not vomit. And plus, they are very novel. So you can see that there are no similar molecules in the database. So very, very low similarity to the known compounds. Currently, we have very, very good under 50 nanomolar inhibitors of SARS-CoV-2 3C-like protease with reasonable medchem properties. So you can see that some of those, like 50 nanomolar, 43 nanomolar, already have reasonable ADME, already have reasonable pharmacological properties, those are novel. So they are generated using AI. Yeah, so far they are, to my knowledge, better than what is published in the literature. And again, those compounds that were published previously in Nature Science Cell, at that time people were rushing to publish, those are older compounds, right? So dating all the way back to like N3 compound that dates back all the way to 2000 or 2005. We have 30 to 50 nanomolar inhibitors that are very novel. And now we are launching this platform for other people to use. So Pandomics allows you to identify targets. So all of you are welcome to apply. Once we release access to this platform, you'll be able to test it. It helps you identify novel targets. Some features of this platform are free. Chemistry 42, that's a system which allows you to very quickly generate small molecules with desired properties. We're deploying this only to select big pharma companies. So currently there is a big lineup of companies who are trying to get access to this system. Some who are, much more legacy companies and don't understand AI, they're still on the fence. But we currently have a very long list of pharma companies who are deploying. And in Clinico, that's currently, we're collaborating using the software with hedge funds. So our aim is to, digitize, disrupt, democratize, and dematerialize drug discovery. So anybody can do it regardless of where they are. But now I'll switch gears. I know actually I'm almost out of time, but we started later. I'll try to cover my favorite topic very, very quickly. And we're switching gears to deep longevity. I'll be very happy to participate in a Q&A session a little bit later over time and help you with any of your career questions. But my biggest challenge and my biggest question is, how do we go after aging? Because I believe that aging is humanity's biggest problem. It's every one of yours biggest problem. So regardless of what you do to the patient on the pharmacological side, if you're curing a disease, they will die, and they will die within their lifespan, right? So the average lifespan in the US is still like 79 point something. In Hong Kong, the city where I'm, China, the city where I'm dialing in from, life expectancy is 86 and a half. So it's the highest in the world, and it's actually significantly higher than the US. So the average Hong Kongese lives 6 years longer than an average American. But still, it's a very small limit, right? So if you think about what preventative medicine can do in addition to treatment medicine, you could push this boundary significantly, right? So a 10% lifespan increase would give you additional 8 years of life, right? So that's a lot. So my life's goal is to identify interventions and diagnostics, how to measure aging, and how to give this, 10% additional lifespan, or hopefully more. Because if you eliminate cancer completely, so there is no cancer now, the average life expectancy is going to increase by 3 to 4 years in general. So it's not going to, result in a very dramatic increase in life expectancy. And so are the many other diseases. So aging is still the greatest problem. It's a huge trend as well. So in pretty much every country, you are going to see many more older people. It's a huge economic problem. So most of this increase in 2008, from 2008 to 2020, is due to aging. Actually, I wrote a book about it. So it's called The Ageless Generation, published by Palgrave Macmillan in 2013. So when I was writing this book, the U.S. national debt was, around $12 trillion. Now it's 27 trillion. Actually, when I was giving this presentation a few weeks ago, it was 26.5 trillion. Today, it's 27 trillion, and it's increasing dramatically. Well, COVID and fiscal imbalance, but primarily due to aging. So healthcare is a huge component of that, and life expectancy increases are a very recent trend. So in the days of ancient Greece, where you are dialing in from, life expectancy was like 30, 35. So, I should not be alive already by those standards. I would be considered to be an ancient man. And only in the 20th century, in the US, in the beginning of the 20th century, life expectancy was 49 and a half. And since then, it has almost doubled due to hygiene, better quality of life, antibiotics, early diagnostics. So it's a very new paradigm. There are many new advances that will significantly extend our life expectancy going forward and increase longevity, but not necessarily productive longevity. Last 10 years, I've switched to aging research in 2004. So 16 years. So 16 years ago, this field seemed pretty barren, so there were not even good books. And in the last decade, many new advances came to life, right? And even CRISPR, for which Jennifer Doudna and Manuel got the Nobel Prizes. Nobel Prize, shared Nobel Prize, which is great. And iPSCs, so Shinai and Iwanaka got a Nobel Prize for that. So very rapid advances in technology. And what I see as one of the major advances in aging research is the advent of aging clocks. So now we can measure aging, and we can predict your biological age and not your chronological age. And that age, biological age, you can predict at many levels, and it will be much more predictive of mortality than your chronological age. That's typical definition. So what we invented at InSilico at that time, and now we passed this IP to Deep Longevity, we were the original inventor and the first guys to publish on this. We created deep aging clocks, where we predict your biological age using many different data types using deep learning. Now we've got some very simple molecules like nicotinamide riboside and NMN, nicotinamide mononucleotide tested for life extension. Metformin is tested, rapamycin is tested. So again, this is a great time to get into this industry. And I'll try to accelerate. So currently, traditional preventative medicine does not really look at you in your entire kind of lifespan context. It looks at you within the age range. Even most of the references are given for the specific age range. And no doctor knows you for your entire life, at least super highly qualified physicians. And what we are trying to do, we're trying to build AI that identifies and monitors this minute transition between age-associated processes and age-associated damage, age-related damage, to age-associated pathologies. So now we don't need to define aging as a disease. AI does it for us. It basically creates this link between pathology and disease. And the way we do it is we take very large longitudinal data sets. So think UK Biobank, but it's only one of the many sources of data that could be used for something like that, where millions of people are monitored during their lifetime, and a lot of different data types are collected. So what we realized in 2015 is that deep neural networks are great at generalizing and identifying very complex patterns in data. And what we started doing is we started creating very complex deep nets, and sometimes very simple deep nets, that have a lot of neurons on the input, where you can put in the features from pretty much any data type you like, especially statics, like, for example, blood tests. So one neuron would be connected to one feature like albumin, glucose, alkaline phosphatase. And you have many less and less neurons per layer in the latent space, and then you are predicting just one feature, age. So trying to predict your biological age. If you are using this data from healthy people, not from cancer patients, you are predicting more or less biological age, so age in the ultimate healthy state. And there are many, many other ways of doing this. So after this network is trained, it can predict mortality, it can predict your biological age, and then depending on what data type you use, it can be used to identify pathways, targets, causal graphs, and even generation of synthetic data types. Because one of my favorite, actually, functions of GANs is the ability to generate an individual with age as a generation condition. So you can make people younger or older in pictures. There are funny apps that do it. But when you are doing this on non-imaging data or more complex imaging data like MRI, CT, or X-ray, or on blood tests or activity, you're generating really, really cool, valuable data that basically projects the patient into the future or into the past. And you can validate with longitudinal data, which is super cool. So Deep Longevity is focused on that. And our idea is to develop this new field of longevity medicine. So we basically, my definition of longevity medicine is the AI-powered preventative medicine, where you would look for new ways to make the patients look younger to the deep neural network with multiple interventions on multiple data types, on blood, on transcriptome, on methylation, on microbiome. And to do this, we need to identify the most important features the change with age, and then look for ways to modulate those features. So I'm going to skip a few slides, but one of the really cool papers that explain this model and hypothesis was published by us. So one of my collaborators, Polina Mamoshina, and myself in Aging. It's called Deep Biomarkers of Aging and Longevity from Research to Applications. And now a lot of people in China picked it up, right? So these guys are great at recognizing very important trends and kind of taking them forward. So there will be more and more research out of China coming out. Hundreds of thousands of people are going to look into this. And I highly encourage you to watch the lecture at our agingpharma.org. So if you go to agingpharma.org, that's the conference that I help co-organize every year. We used to do it on Basel, but this year we did it online. And next year it's going to be in Columbia University in New York, hopefully. So this year it broke all the records, 2.6 thousand people, mostly PhDs, MDs, who participated in this. And here you can see, the speakers and one of the important lectures to watch that I highly recommend. Of course, I recommend all of them, but this one, Dr. Li Kai-Fu Li, AI and Longevity, he's probably the most influential person in AI in the world because he is #1 AI guy in China, has like 70 million followers on WeChat. And last time I did a drug discovery lecture, very boring lecture with him online. It went and got 10 million views in 48 hours. So that's how popular drug discovery is in China. So all of you are going to be competing against or collaborating with the brilliant scientists in China who are, right now it's Sunday and a lot of people are sending me emails now, WeChat messages, they're working. So one of the great scientists who talks about AI and longevity is Kai-Fu Li. I highly recommend looking at that lecture. Again, following this Sci-Innovation Fund, it's a wonderful fund, which, and also the AI Institute by Sci-Innovation, they have about like 1.2 thousand AI scientists looking at many areas. And this paper describes, the basic applications of deep neural networks in drug discovery, in aging. So what we did, we trained deep neural networks to predict biological age using many different data types. So some of the data types are microbiome, transcriptome. We got US granted patents on those. Hematological aging clocks, so just very basic blood tests. So if you want to try it out, you can download Young AI app or go to Young AI web, key in your latest blood test in a more or less anonymized fashion. Don't use your real name, and see how it predicts your biological age. And we have also methylation aging clocks using deep learning, photo aging, behavioral aging clocks, many different aging clocks. And what we did, we also were clock agnostic. So it also kind of tells you about the value of different data types. So some of the data types, like photographic picture, can tell a doctor quite a bit about the patient, sometimes more than the patient's genome, because it tells you the patient's age, body composition, some very basic phenotypic characteristics, even sometimes the social status, right? Depending on how the patient is dressed in the picture. The genome cannot tell you that. So we are data agnostic. Some of the data that we are using for aging clocks are very simple surveys, but then you get into more complex data types, and you can get transcriptomics, proteomics, microbiomics, and get data from wearables and predict the age. And in Young AI Web, you can see how those predicted ages change in time in response to the various interventions or lifestyle modifications or lifestyle choices. And see how patients change in response to the various drugs. So for that, we also create Longevity Cloud, so where you can upload the data from the patient, very simple anonymized data, to predict the age before and after the various interventions. And we also have a mobile app. Just went into App Store, only available for iPhone right now because we want to have a controlled environment. and we are testing it right now with many clinics. Human Longevity, a company which kind of provides very, very sophisticated personalized diagnostics for primarily ultra high net worth and high net worth individuals who are willing to pay a lot of money for that. They're in San Diego. It's Craig Venter's company. You know, Craig Venter, one of the top human genome pioneers. So they do MRI, whole body, body composition, core labs, medical history, cardiac MRI, CT, and expanded panel of blood tests. What we are doing is we are providing aging clocks in the context of this data to see what can be changed to make people a little bit closer to their chronological age or even younger. So that's the idea. And again, we recently started going into psychology of aging. So I have a paper coming up which predicts age using the various surveys that are not directly related to your chronological age, to see, you know, how old you are psychologically, because the older you, the longer you expect to live, the younger you are going to behave. So that's actually another really cool feature to learn today. And yeah, so I have over 130 people right now who are contributing to this effort. We're looking for new people. We're looking for you know, great talent worldwide. And of course, we're very collaborative. And many of them are very Greece friendly, so it's very easy for them to travel there or accept people from Greece. So thank you from Deep Longevity, and thank you from InSilico Medicine. You can find me on WeChat, and here is my email. Not necessarily I'm going to respond quickly, but I try to respond to emails.
Thank you, Mr. Zavoronkov. It was so interesting and insightful, and I can reassure you that you weren't boring in this online talk.